Okay, so this is one. When you do this practice question, mark yourself as slides what is corresponding to that. Then you could review not only this question, because you could review what is amphitrichos, what is lithotrichos, what is petrotrichos. It's all right there. Then you can get a good idea what are they. You know. Koch postulate is easy, so you can go back to see the slides. The slides is back in the. That's when we talk. All the end is lecture 1. Is that right? We talk about Koch's postulate, and I even draw on the blackboard some of the simple explanation, but the postulate is right here. And to give you example is tuberculosis. Microbacterial tuberculosis is in guinea pig. So that's a Koch postulate is right there, 1, 2, 3, 4. So that one is not difficult at all. So let's see other questions we miss. Okay, let's look at the 53 question. There are examples of prokaryotes that are resistant to the antibiotic penicillin. Provide one simple explanation for how microbes could be resistant to penicillin. This question, I said, is only one sentence. What is that? How you get it? Because, is that plasmid? Resistant plasmid. The plasmid of an antibiotic-resistant bacteria could be directly taken by another bacteria, we call it a transformation. It could be going through a phage, we call it a transduction, and it could going through a pili, P-I-L-I, a pili, sex pili, that is called a conjugation. It all comes from all plasmid. So what is plasmid? It's a single around 30 base pair short BP, circulated most of the time, could be linear DNA. It could do the replication independently from the chromosome, or it could integrate the DNA sequencing into the chromosome. Is that right? That's a very simple answer. If you don't know, we go back to look at the slides again. Right here, talk about the plasmids. So that question is, this slide solves the problem. When you do this question, go back to read it. Then all the key points is there for the plasmid. Answer clearly is right here. All plasmid. So simple. That's why I said using only one sentence, you can solve the problem. Okay, let's go up to C. Look at this question. Okay, the energy required for transport via ABC transport is derived from. What is ABC? Is ATP binding cassette transporter, and I wrote on this corner of the blackboard. That's ATP hydrolysis. Is that right? It's so simple. That's ATP hydrolysis. When grow a bacteria or blood agar, a clear zone around the colony of Streptococcus pyogenes indicate alpha, beta, gamma is a beta hemolytic. I want you to know this because we were talking in examination 2. Lots of the pathogen, the characteristics, and this is a beta hemolytic. And don't confuse with alpha hemolytic. And I show you the slides already. That's a key important slides. So here we talk about already. Okay, let's look at here. Which of these methods can be used to determine the number of viable microorganisms in a sample? Okay, this is the key. Viable microorganisms. Viable microorganisms, which means we need to know the methods testing is a live cell. So which methods we should do? Can we do lighter scanner in a spectrum photometer? We know the whole population, but we don't know the live and dead because it's all there. Can we measure cell mass? It's the same thing. We don't know the live and dead. So measuring colony forming units, CFU, is that correct? Yes. Z is 1, we spread plating or pore plating on the other. So the answer is C. Counting unknown volume cells in a hemocytometer, this is just confuse you, and I don't think it's related to this question at all. Most bacterial cells divided by what? Binary fission, is that right? We talk about doubling time binary fission. And I forgot to mention in the lecture, they should look exactly the same, but most of the time it's not because of the mutations. That happens all the time. Okay, we mentioned about this already. Okay. This question here, we did this label. You have to go back to permease proteins that aid in the transport of nutrients resemble enzymes in their specificity for the substance or transported. Each carrier is selective and will transport only a close related set of substance. Is this true or false? This is tells you the concept of the enzyme. This enzyme usually is targeted for one substrate. So the answer is true. It's only one type of the carrier will do. It's not have a universal carrier for that. The movements of molecules from an area of high concentration to an area of low concentration without participation of specific carrier is called what? You can do this in biology 101, is that right? Passive diffusion. It's simple. Bacterial ribosome, known as what? 70s, 50 combined with 30 is not add up with 80. Because S is a Vandenberg unit. It's a speed precipitation measurements. It is not to measure the molecular weight. Don't forget about this. Always remember. All of the following statements about the Paris plasmic space are true. It is found only in gram-positive bacteria. That is not correct. It is located between the plasma membrane and the outer membrane of gram-negative bacteria. That's not really correct. It contains hydrolytic enzymes and binding proteins involved in nutrition acquisition. It should be C, is that right? Yeah. Except. Which is true, except it should be C, yes. Lipopolysaccharides we mentioned. We mentioned this already. Small circular DNA molecules capable of replicating and containing genes that are useful but not necessary to bacteria called what? Yeah, plasmids we just mentioned. What is the name for the cell wall model? Fluid mosaic model. Okay. Endospore has a calcium diproconic acid. This is easy. Okay, let's look at this question. 25. You discover a new transport system used by newly discovered bacterial species. The sugars that are transported using this system are phosphorylated as they enter in the bacterial cell. What we call this? Remember in the classroom I said there is a passive diffusion, facilitated diffusion, and active transport. None of them actually is really for bacteria, although we talk about that all the time. The major thing for bacteria is group translocation. So if you forgot about it, go back to look at your slides. It should be in the group translocation should be right here. When we talk about this, the intake of the bacterial culture is right here is group translocation. There is a multiple group helper for help the phosphates. The idea is that this glucose mannitol could directly become a mannitol 1-phosphate, glucose 6-phosphates. And these you will see there's energy potential because of the phosphates. So they were directly involved into glycolysis or modified glycolysis. So we'll make the energy using is more efficient. So that's why the group translocation is most likely to be using by the bacteria. So that's why here the system is group translocation. Okay. Which of the following processes can be used to bring nutrients into the cell against the concentration gradient? What means against? From low to high. So active transport and the facilitated diffusion. 22 chemotaxis. Moving toward attract or move away from the repel. I said it's a round trip. So A is the answer. We already talked about this. Most of these we talk about. And the beginning is some of the history. That is easy. Okay. Okay, so we all have this stuff. Exam 1, usually everybody can do very well. The key for get A in this class is exam 2 and 3. Because exam 2, we're going to move on to the clinical microbiology area. There's lots of the bacteria name going to suddenly come to you. And the examination 3 is genetics and biochemistry combination. Exam 1, most of you should do very well. So I hope you study, do these practice questions, go over the slides, do very well in the exam 1. Okay?